afternoon, Beth. Long time no see.
Yeah, hi Abu. How are you today?
I am doing great. It's been, I think this is probably my fourth bio. How many bios have you done?
I think probably on and off about 5 or 6.
Five or six.
Yeah,
That's awesome.
yeah.
Well,
It's a feat.
it is a fun one to do. And yeah, so we're recording live at the Biolabs booth,
Yeah.
Bio 2026 in San Diego. And as part of that, it's our Biolabs b.empowered podcast. Yeah, so my name is Abu. I am the site head in San Diego. You and I know each other a little bit, but I'm sure the audience don't know much about us, so we can, get it started and then, yeah, introduce ourselves a little bit.
Very
So,
good.
just to begin, you work and you run Origami Therapeutics. Can you tell us a little bit about that and, what your
Sure.
origin story?
Sure. So I'm Beth Hoffman. I'm the founder and CEO of Origami Therapeutics. So my background is originally as a cell and molecular biologist, but I've been in the pharma industry for over 25 years.
Okay.
So worked at places like Lilly and Amgen and Vertex,
Okay.
and really so it was my experience at Vertex that led me to decide that I would start a company.
Okay, well, that's wonderful. I mean, not everyone who comes up with an idea ends up starting it. Sometimes it is for financial reasons, sometimes for execution reason. So I was just wondering, in your case, how does being embedded in the Biolabs ecosystem help, maybe hamper? I would like to
Yeah.
know about
So
that.
so I'll say we were at another incubator whose
Okay.
name I won't mention.
Okay.
And so what attracted us to come in and join Biolabs really was the setup, so the benches, but also for us, the shared equipment. And this was really critical because what we do a number of different techniques, a lot of cell biology, biochemistry, molecular biology, and so we really need both the cell culture facilities as well as a lot of equipment.
Got it.
So that became really, in a way, a no-brainer
Yeah.
that Biolabs was the right place for us. And that's what we thought coming in. And I think over the past 3 years, that's really been borne out. So as I'm sure people know, you end up having to do new techniques and new things and need different equipment as the research and the drug development unfolds. And so that's really what's available at Biolabs.
So it's help, not hampered. I'm happy to hear that. As a site head, you always want to be able to be on the other side of solving a problem,
Yeah.
not creating one. And so, and speaking of problems, I mean, you're working on neurodegenerative diseases. It's a global, issue, right?
Yes.
Affects a lot of people, different backgrounds, different nations. So it's an ambitious goal to say, I am gonna go get started and solve that problem. So, what led you to actually jump into a problem such as that? And actually, you can tell us the actual indication because I just went general as to.
Sure. Twofold. So most of my experience in industry has been in the neuro space.
Okay.
Even though my training is in cell molecular biology and very much looking towards the translation between the preclinical and the clinical work. And so my experience at Vertex was working on the cystic fibrosis drugs.
Yeah.
And so the commonality, and so we've really been leveraging what I learned at Vertex working on these successfully launched drugs that have transformed the way patients live their lives. And so that's what I wanted to do for neurodegeneration. And the commonality is that we work on small molecules,
Okay.
and these small molecules can change the way proteins fold. And for proteins, if you don't have the right shape and you don't fold correctly, you don't function correctly.
Yes.
And sometimes you will lose function, and sometimes you will gain toxic function. In the case of neurodegeneration, a lot of the times it's about a gain of toxic function,
Oh,
right?
so gain of function is not always a good thing.
It's not always a good thing.
Okay.
So, sometimes these molecules, when they misfold,
Yeah.
they talk and play with other proteins and other components of the cell
Yeah.
that they're not supposed to talk to.
Oh, gotcha.
Other times they actually are toxic because they inhibit other important cellular processes. So in the case of But why did we embark on this? We did this in a way that we wanted to be sure that we were um, de-risking as much as possible. And so, much like we did for cystic fibrosis at Vertex, we've focused on monogenetic forms of neurodegeneration. So we know what's broken, we know what we need to fix, and then we also will know who to recruit into clinical trials. So I've put more than 30 compounds in the clinic, most of which have not been successful. That's not unusual. But I wanted to skew the odds in our favor because neurodegeneration has been such a challenging field. And so that's what we did. We picked Huntington's disease. I had worked on it before. And then what we did was we did a phenotypic screen so we could, and we measure what is clinically relevant. So exactly what we want to measure in patients down the road in phase 1 and phase 2.
Wow.
Okay. And so in that way we were able to, I mean, that's really what motivated me to start the company, is an idea where I think we sufficiently de-risked what we were doing to our favor.
Got it.
So we were trying to optimize the probability of technical success.
Nice. So if I understand correctly, monogenic disease, you have your genotype to phenotype, some gain of function issues that you really didn't want to, but you ended up like, hey, we need to come up with a way to figure out misfolded proteins, which are core to most diseases, but especially neurodegenerative diseases.
Many. And in the end, we have small molecules that cause the degradation of the mutant Huntington selectively. And so this makes us quite differentiated. And in fact, we are lucky enough to form a collaboration with a pharma, Ipsen, and so that really has allowed us to expand what we're doing, and now we have an opportunity to build our pipeline.
No, you actually segued to the next question, which is, known you for about a year and a half now, through biolabs, and recently there was some news right around JPM. And we couldn't help ourselves but share with everyone on LinkedIn. Can you tell us a little bit more about that story that was shared earlier on this year?
Sure. So we signed a deal at the end of January. It's a collaboration initially on a preclinical asset, and
with whom?
With Ipsen,
Ipsen, okay.
And a French company, and then once we get to a certain stage, they will have the option to license. So it's a typical biotech deal with milestones and commercialization and royalties, etc. But interestingly enough, they asked us to meet with them at Last Bio in Boston.
Okay.
So here we are a year later, and things went pretty quickly. And they've been a great partner so far, and I think they're really good for a small startup like ourselves. And so they're interested in both rare disease and neurodegeneration, and so that really we fit with what they were looking for.
Well, that's awesome. So I've spoken to quite a few founders at Biolabs, some of whom were just starting out of right out of grad school, out of postdoc, and so on. And from what you're telling me, I mean, it's a few scenario where folks think about, all right, here's what my clinical endpoint would be. I am going to start a company to make sure I get to that goal. So let's crank the wheel of time backwards and say, what advice would you have given to yourself from way back time if you were to start a company like Origami? Because the kind of things you've just told me aren't the things that someone would just jump right in as a first-time founder.
That's true.
Yeah.
But so since I'd been in pharma, I had a really good idea, and a lot of what I did was exactly what we're doing, right? A lot on the preclinical side, and so so basically, I guess I had some confidence of what could be. What would I? So I think we've done really actually surprisingly well. I mean, better than I perhaps initially anticipated. But, also because I had done a lot of this before, I felt like, okay, let's jump in, let's get it done. If I wait till I get a lot of funding and, socialize this with a number of VCs, etc., that's just time wasted.
Yeah.
I probably could have done a little bit more of that, but we were really fortunate to get some SBIR funding, a couple of Kickstarter awards from O2H Discovery to help us with the chemistry. And so in the end, when I look back, I think we are beginning to accomplish what we set out to do, and a lot of people these days go after don't go after small molecules,
Yeah.
and we went after small molecules because what I tell my people all the time is the patients and their families are waiting. This is we have to. It is a systemic disease,
Yeah.
and we want to hit all the cells in the brain and the rest of the body, and that means we need to use a small molecule. So.
Patients and their families are waiting, and you
That's
don't want
right.
to keep them longer. No, I
Right.
think that's
Yeah.
probably a
And
great place to end. I'm really grateful for this. Thank you very much for sharing the story with us. Wish
thank
you the
you.
best of luck, and I want to
Thanks.
see the team really grow and do some cool
And,
science.
and we have, and
Yeah,
so and we're grateful to Biolabs for having additional space that where we could expand very quickly.
it's our
So
pleasure. All right, ladies and
thank you.
gentlemen, that's the story.
Thanks so much.
Cheers.